Hi, everybody. Thank you for joining us today. Today's webinar in the Clinical Trials Methodology course 2016 series. The topic for today's presentation is what you need to know before talking to your statistician about sample size. Everyone but our presenter today will be muted. This is to prevent any feedback during the presentation, but we would like the webinar to be interactive, so we do encourage questions. If you do have a question, you can use the Request to Interact Live button. It's at the top right, and it kind of looks like a hand. If you press that, I can grant you the microphone in order for you to ask your question. There is a 5-second delay when you do get the microphone, but after that, everyone will be able to hear you, and if you have your video camera on, everyone will be able to see you as well. You can use the Q&A box to also type in questions. There's a event chat box. It looks like a speech bubble at the top right under the hand button. And that contains a link that you can download for discussion later on in the webinar. If you're interested in CMEs, you must complete an evaluation form. There'll be details at the end of the webinar regarding this, but you should also be able to see a little tagline that's broadcasted across your screen, and that's the link you're going to use. So I'm very pleased to introduce our speaker today. Dr. Yeatts is an associate professor of biostatistics at the Medical University of South Carolina. She's a faculty member with the Data Coordination Unit, an NIH-funded statistical and data coordinating center that specializes in the design and coordination of multi-center clinical trials. As the PI of the Statistics and Data Management Center for the Phase 1 and 2 trials for deferoxamine and ICH, Dr. Yeatts is responsible for the design of the trial and the implementation of the statistical and data project management work scope. She's also the PI of the National Data Management Center for DIFFUSE-3, which is a multi-center clinical trial using adaptive design to assess the efficacy of endovascular therapy following imaging evaluation in ischemic stroke. In addition, she's also the primary unblinded statistician for the Interventional Management of Stroke Trial IMS-3 and the Progesterone and Traumatic Brain Injury Trial, which is PROTECT. As a co-investigator on these grants, she was responsible for the statistical monitoring of data and the implementation of interim and final efficacy and safety analysis. She also serves as data and safety monitoring boards and is a grant reviewer for several funding agencies. Her primary research interests include the development and implementation of efficient early phase trial designs and novel trial outcomes. Thank you for joining us today, Dr. Yeatts.
Thanks, Jenna. I always think those introductions are awkward in person, and they're even worse on the webinar. So thank you all for listening to all of that. So I am going to talk to you a little bit today about the factors that go into determining sample size when you're meeting with your statistical collaborator. I will start with the disclosures that are required, since you all, some of you will be getting CME credit. I am on the steering committee for the PRISMS trial, and I do receive consultant fees from Genentech for that role. But one of the nice things about being a statistician, I don't really have any interest in talking about unlabeled use of commercial products or investigational use. So this presentation is not going to include any of that. Now, it is noon on a Friday, and I know that's a hard time for a lecture, and it's a really hard time for a lecture on statistics. I hope that we will keep this light and informative, and feel free to ask questions as you have them. But I am going to start out by showing you sort of a tongue-in-cheek example of what not to do, or how not to go about that conversation with your statistical collaborator. There are lots of these videos on YouTube. If you haven't seen them, I encourage you, when you need a laugh, to take a look. But I'm going to play this one, which basically is a representation of the type of interaction that I have had and many of my statistical colleagues have had with our investigators, who maybe when we first get to talk to them just don't get how complicated this discussion might be. Hopefully this will play.
I have a study, and I need to know how many patients I need. I think I only need 3 patients.
Okay, what kind of study is it?
I'm doing a lab study. Can I just use 3 patients?
That depends. What are you trying to show?
We always use 3 patients.
Okay, what are you trying to show?
I'm trying to show that drug A and drug B are better than only drug A.
How are you measuring if it is better?
We are looking at apoptosis. Can we just use 3 patients?
What kind of measure of apoptosis? Do you know how variable that measure is?
We always use 3 patients. That is what we have published before.
3 patients per group or in total?
Yes.
Okay, let's try something different. What type of measure are you using? And is this an in vitro study? An in vivo mouse study?
It is an in vitro study of patient samples, and we are looking at measures of apoptosis after treatment with drug A and drug B versus drug A and control, and drug B alone.
Oh, how many groups are you analyzing altogether then?
My grant is due tomorrow. I just need to know if I can just use 3 patients per group.
Okay, well, you probably should have come to me earlier for help with this.
All I need you to do is tell me that 3 patients are okay.
So, it is not that easy. Your marker for apoptosis may have a large amount of variability associated with it that could make it difficult to ascertain differences based only on 3 patients per group. It also depends on what kind of outcomes you are looking at. Are you looking at normalized changes in expression levels from baseline? 3 patients may not be sufficient to detect differences in these expression levels between the different treatment groups.
I do not understand.
I'm saying that you may need more than 3 patients per group, but that I need more information to determine that.
The serine protease granzyme B is made in cytotoxic lymphocytes, where it is inactive due to the low pH of the storage granules. It is measured immediately upon delivery into target cell cytoplasms, where the pH is neutral. This is followed by cleavage of process phase 3 by granzyme B in the target cell cytoplasm and the subsequent induction of apoptosis. All proteolytic events can be quantitated at the live single-cell level by flow cytometry and imaged by optical microscopy.
I understand very little of what you just said. Maybe we should talk more about this.
I just need to know if 3 patients are enough. My grant is due tomorrow.
We will need to meet to better understand this before we can determine the appropriate sample size.
I can put you on my grant for 0.5% FTE. You can design and analyze the data later.
Do you really think that is enough to do the work?
Well, it may be less if it is awarded and the budget is cut.
Please go away.
Okay, so of course, none of the statistical folks that you'll meet through this course or hopefully at your institutions would ever tell you to please go away. But again, these are the sorts of conversations that we sometimes have with investigators. So I do hope that what you'll get from this talk is sort of a sense as to all of the components that go into this discussion. But perhaps even more importantly is an understanding that it's not likely to be a 2-minute answer for most experimental designs, unless you've been working with a particular collaborator for a lengthy period of time and they're already familiar with the disease area and the types of experiments that you do.